recording. Just tell very good. I see the signal. Very good. So I'm now able to begin. All right. So what we're last class we went through some basic things that I wanted you to know about the cortex. And so what I'm now basically going to do is continue this lecture onto the cerebellum. So again, just like I've said before, in most cases, whenever we're looking at a structure and trying to examine its function, very often we're basically looking at that structure in terms of groups of cells that have receptors and have dendrites, and then whether the cell fires or not is then going to be produced in the axon and to its projection point. So, okay, two people are coming in. Okay, so, okay, I see John Lyle coming in. Okay. Okay, so in any case, the cerebellar layers, John, Matthew Rowland is recording this lecture.
Oh, wonderful. He's a true legend.
Okay, very good. So that's what's happening. So in any case, for many, many structures, what we basically see is that particular area getting inputs and then sending outputs. The three major areas of the brain that operate in a subdivision kind of way, of course, is the hippocampus with its 6 layers, the cortex with its 6 layers, and then the cerebellum outside of either the dentate nucleus or the deep cerebellar nuclei in the vermis. The rest of the cerebellum operates in a layer-specific type of way. So when we look at cerebellar layers, we basically see 3 of them. One is the molecular layer, that is the outermost layer. Then 2 is the Purkinje layer, that's the middle layer. And then 3 is the granular layer, which is the deepest of the cerebellar layers. So the major output of the cerebellum comes through Purkinje cells, and Purkinje cells, as you can see, are in layer 2. And like their counterparts, the so-called pyramidal cells or Bet cells, these Purkinje cells are quite large, and what they have, again, is a rather elaborate set of dendrites. And these dendrites extend upwards into the molecular layer. So, communication about how these Purkinje cells should be activated or not activated will depend upon the excitatory or inhibitory input coming into these Purkinje cells. Now, there's a major difference between cerebellar output and cortical output. If you paid attention, you basically saw that an enormous amount of cortical output, whether it is going down into, going onto the striatum, going back onto the thalamus, going on to structures in the limbic system and then going further on down, a very large amount of this output is glutamatergic and therefore excitatory. So it's basically activating different parts of the brain. The cerebellum with the Purkinje outputs, by and large, are inhibitory. So about 99% of Purkinje outputs are GABAergic, and therefore will inhibit whatever projections or contacts that they make. And of course, this then becomes a very important kind of thing of when you sort of do a quick word association of the cerebellum, that very often the single word that sort of pops up whenever you say cerebellum is coordination. And coordination basically means, even on a very, very simple level, is a series of motor outputs that flow over time. We may think of it as a response. I mean, psychologists are very, especially animal psychologists, a rat presses a lever once. Well, if you just basically think of a rat pressing a lever or doing a nose poke in order to get a response, that is an actual coordinated response that needs an awful lot of muscles. That what you basically need to know is where the organism is relative to the thing that's responding to, and then it has to go and do all of this. And this is what the cerebellum is basically doing. And a lot of it is done not through excitation, but rather it is done through, if you want something activated through disinhibition. That is inhibiting an inhibitory synapse. So we now look at these Purkinje cells and recognize that the Purkinje cells are getting inputs largely from the molecular layer. And the molecular layer is made up of things called parallel fibers and then stellate and basket cells in the Purkinje layer. And those stellate and basket cells very often have short appositional synapses onto the dendrites of Purkinje cells. But where do the parallel fibers come from? Well, the parallel fibers actually emerge from the granular layer, layer 3. Notice that there are 3 types of cells in layer 3: Golgi neurons, granule cells, and cerebellar glomeruli. What each of those types of cells do is well described in your book, and you can look at the neurophysiological properties. But one of the things that I want to do for this lecture is basically start to understand in some way how these 3 layers interact. And the best way of doing this is to use a single type of input. And that single type of input comes from a set of cells that are described when we move through the medulla. We noticed in the caudal all the way up to the rostral medulla a rather large series of structures that took up the ventrolateral part of the medulla. And that was called the inferior olivary complex, which is made up of the principal nucleus of the inferior olivary complex, the medial accessory, and the dorsal accessory nuclei. Now, especially in the inferior olivary complex to principal nucleus, there are two major forms of fibers that emerge from that principal nucleus. One is called climbing fibers, and they basically look like an ivy vine climbing up a wall. So I very often think of Wrigley Field, the baseball stadium that has all of those vines. And the climbing fibers coming from the inferior olivary complex, principal nucleus, come up, and they basically synapse in the dendrites of the Purkinje cells. And usually what they do is one particular climbing fiber may go and synapse on a variety of Purkinje cell dendrites, a number of them. And what the function of those climbing fibers on these Purkinje cell dendrites is typically excitatory. So you're basically getting the firing of climbing fibers. So you then can ask, what about that climbing fiber, the cell in the principal nucleus of the inferior olivary complex? So that particular cell that is there can either be excited or inhibited. Very often it is excited by spino-reticulo fibers that are coming up from the spinal cord, from the different levels of the spinal cord. And this, and very often the type of information that is coming in there is what we would basically call proprioceptive information. And what proprioceptive information is, of course, is not just sheer sensory input, but sensory feedback input. So, and that then fits into that schema when we look up the cerebellum and coordination, because what you're basically seeing is this continued feedback as the organism may be making a complex response, like moving a limb through space, etc., in some smooth kind of way. On the other hand, what you can also get coming down is the cortico-bulbar system. Remember, we took output coming out of the precentral gyrus, the primary motor cortex, and divided that output into two main categories. One, which is corticospinal output, that basically is going down to a specific part of the spinal cord and going into ventral horn, eventually into ventral horn neurons to control a motor movement. But there's a second major pathway that comes out of the precentral gyrus that we have called the corticobulbar tract. And this corticobulbar tract will now synapse in the very specific parts of the brainstem. One, of course, is a series of synapses on the red nucleus to control rubrospinal modulation. A second is to synapse on cells in a reticular formation to control reticulospinal activation. And then you can have corticobulbar fibers that synapse on a substantia nigra that in turn will control a neostriatal function. But a fourth place where it can synapse is that the corticobulbar system can come down and do blanket types of inhibitory modulation of the principal nucleus of the inferior olivary complex. So there's a yin and a yang between spino-reticular excitatory input of the inferior olivary complex and descending inhibitory input. So in any case, the climbing fibers are coming up, synapsing on a number of Purkinje cells and producing this excitation. There is a second type of morphologically identified cell in the principal nucleus of the inferior olivary complex. That is called the Mossy Fiber. So rather than having this sort of ivy-shaped thing, you have a sort of mossy kind of connection, almost like a little bit like a perineural net or some type of reticulum. And what that does, those cells go up and they synapse in layer 3 in the Golgi neurons and the cerebellar glomeruli of the granular layer of the cerebellum. And what those Mossy fibers then do is then modulate the activity of the Golgi neurons and cerebellar glomeruli. Now, where do the axons of the Golgi neurons and the cerebellar glomeruli go? The axons come up out of the granular layer and then go up into the molecular layer. And they are the vast majority of what is now called the parallel fibers. So these parallel fibers now run along the whole base of layer 1 of the cerebellum. And what they do is typically produce a blanket amount of inhibition. They are primarily inhibitory. So what are they doing? They are acting on the dendrites of the Purkinje cells, producing inhibitory postsynaptic potentials and preventing those Purkinje cells from firing. So what you're basically doing is the actions, if you basically think it in a relatively simple, straightforward way, the inferior olivary complex principal nucleus climbing fibers are going to excite Purkinje cells and therefore produce very sort of specific bursts of inhibitory activity. Whereas the Mossy fibers coming out of the inferior olivary complex principal nucleus is coming up and acting on the cerebellar glomeruli and Golgi cells to produce a blanket inhibition of the Purkinje layer through the parallel fibers. So you have a very exquisite degree of modulation. Now, of course, Purkinje cells are also going to be affected by the major inputs coming in from the inferior cerebellar peduncle. And the inferior cerebellar peduncle, if we remember, is made up of posterior spinocerebellar fibers and cuneo-cerebellar fibers, in addition to the olivocerebellar fiber. So inputs coming from, and what does the posterior spinocerebellar tract carry? It carries largely lumbar and thoracic inputs into from the lumbar and thoracic cord up into the cerebellum. Further, the cuneo-cerebellar tract will bring up inputs coming directly from the cervical level of the spinal cord. Then the olivocerebellar tract is carrying the what? The Mossy fibers and the climbing fibers, with the Mossy fibers going into layer 3 and the climbing fibers going to layer 1 and acting on the dendrites of the Purkinje cells. So you have that modulation. Further, what we know is the largest of the peduncles, of course, is the middle cerebellar peduncle. And the middle cerebellar peduncle is again a two-way street. It is fibers exiting out of the cerebellum and coming down and synapsing in that sort of very large amorphous area called the Grisium pontus. And in turn, the Grisium pontus, that's modulated not only by spinal reticular fibers, but also cortico-bulbar fibers, are sending reciprocal fibers back up through the middle cerebellar peduncle. And then finally, what we know is that the superior cerebellar peduncle is going to have, from the different cerebellar layers, but most importantly, from the deep cerebellar nuclei of the vermis, as well as the dentate nucleus of the cerebellum, there's going to be fibers exiting the cerebellum and going up into the midbrain, diencephalon and telencephalon, and in turn, there are reciprocal fibers coming down, as well as that anterior spinocerebellar tract that we talked about. So we have this incredible degree of modulation using the three cerebellar layers. So here is sort of a diagram of the thing, and what you can see is a Purkinje cell, like its counterpart, the pyramidal cell of the cortex, sort of sits in a central place, a very large cell body. But unlike the pyramidal cell that typically has a apical dendrite with branches at the top, you can see that the Purkinje cell usually has a whole, it looks bushy rather than tree-like. So there's an enormous amount of places where you can get synapses of these Purkinje cells. Also, what you have is a very large kind of axon that's going down into the deep cerebellar nuclei. So now what you can also see is you can see the granular layer and the molecular layer. So what you can now see is climbing fibers. And whereas the Purkinje cell axon is going down into the deep cerebellar nuclei, the climbing fibers coming through the inferior cerebellar peduncle make direct connections onto the dendrites of the Purkinje cells. Notice, on the other hand, the Mossy fibers come in and synapse on either the cerebellar glomeruli or Golgi cells, and then the outputs of the Golgi cells, the cerebellar glomeruli, then form the parallel fibers that now synapse on Purkinje cells. And this type of synapse is largely inhibitory on the Purkinje cell. So what we can see is the layers, the granular layer. Most of the inputs that are coming into the granular layer are typically glutamatergic. And then what we can also see is the major output of Purkinje cells is typically GABAergic. And then we have these two different types of afferent fibers, Mossy fibers and climbing fibers. So notice here that each Purkinje cell, so now we're going to get large here, basically a given Purkinje cell may receive input from anywhere from 200,000 to 1 million granule cells via the parallel fibers. And then each Purkinje cell receives input from a number of 500 synapses from an average climbing fiber. And any given climbing fiber innervates anywhere from 1 to 10 Purkinje cells. And then Mossy fibers innervate several granule cells, and each granule cell receives input from several Mossy fibers. So what there really is a sort of a sort of generalized amplification. You know, we don't have lots and lots of one-on-one coordination here. Rather, it's a large, what you can basically use is another interesting neurophysiological term. The neurophysiological term I'm thinking of is tone. And when we talk about tone, what we're basically doing is either increasing or decreasing in a rather large kind of way, whether we're increasing or decreasing excitation or increasing and decreasing inhibition. And that's basically how this very, very complex and large cerebellar structure begins to work. So here we can see that you can parallel fibers sometimes can directly excite Purkinje cells, but indirectly inhibit Purkinje cells through the stellate basket, and that there's feedback through the inferior olivary complex and the Golgi cells. So here you basically see the afferent neurons of the climbing fiber of the Mossy fibers working, and here you can see climbing fibers coming up to the dendrites of the Purkinje cells. So now we start to think about the deep cerebellar nuclei, because what you can see is here is where does the deep cerebellar nuclei get most of its output? Most of its output is coming from Purkinje cells. And they're coming and they are typically inhibiting these deep cerebellar nuclei. Whereas when we look at Mossy fibers coming in, what you basically see is a lot of simple spiking of granule cells that then form the parallel fibers, and Golgi cells that come up and work on these parallel fibers and the dendrites of the Purkinje cells. So again, another drawing of this. You're seeing the climbing fiber coming up onto the dendrites of the Purkinje cells from the inferior olivary complex. And what you're basically seeing is the Mossy fibers coming up on the granular cells and then modulating the Purkinje cell dendrites through the parallel fibers. And then the Purkinje cell comes down and will now work on a deep cerebellar nuclei, and it'll either work through excitation via Mossy fibers or inhibition. And in this slide, I'm just not going to go through this that much because it really gets into Beeler's area of the neurophysiology, is that what you can see is that climbing fiber action potentials basically produce activation and excitation through calcium conductance and prolonged depolarization, producing a complex spike. And then Mossy fibers basically work through simple spikes in Purkinje cells, whereas climbing fibers work through complex spikes in Purkinje cells. Okay. So probably one of the people, and again, a drawing like this was done by Ramon E. Cajal. And Cajal was one of the first people to really identify, other than Purkinje cells, because Purkinje cells are named after Purkinje. But what Cahal basically was able to do is basically show you the sort of complex overlapping dendritology of different types of Purkinje cells. And what I'm always happy to tell you is that the type of microscope that Cahal would typically work with when he was doing all of this unbelievable, groundbreaking artistry and microscopy was basically equivalent to the type of microscope that one might find in a 5th or 6th grade science classroom. A lot of times basically used mirrors in order to put the light through the microscopy. And of course, that did not deter him from being the unbelievable artist that he is and whatever. And again, I was very fortunate a couple of years ago when I was out at University of Minnesota. It corresponded with the time where the Institut de Cajal in Spain released all of his work and sent it on a world tour, and basically looking at the unbelievable accuracy and beauty of the brain. So now the other thing that we can basically do about the cerebellum is basically look at this, and this is looking at it in a horizontal section. So what you're basically seeing up here is rostral, down here is caudal, obviously medial and lateral. So what we basically see is, first of all, cerebellar hemispheres on the left and the right, and then we see the deep cerebellar nuclei that is defined as the vermis. And the cerebellum is sort of subdivided to 3 main areas because of the presence of 2 very primary fissures, the primary fissure and the posterior fissure. There is a 3rd fissure that sort of subdivides the posterior lobe, called the horizontal fissure. So rostral to the primary fissure in the cerebellum is the anterior lobe that contains the vermis. Then between the primary fissure and the posterior fissure, we have the posterior lobe of the cerebellum, and that's sort of subdivided into an anterior and posterior branch by the horizontal fissure. Then the posterior lobe is separated from yet another small lobe by the posterior fissure. And that 3rd lobe has the sort of wild name of the flocculonodular lobe. And again, you can go through your book and reading what the different functions of the anterior, posterior, and flocculonodular lobe are doing. So when we look at motor and cognitive functions, what we can basically see is 3 types of input. One type of input is spinocerebellar input. So what are you getting from there? The classic somatosensory proprioception from the spinal cord. And where is that coming from? That, of course, is coming from the nuclei of Clark, as well as cervical nuclei in about layer 6 that then form the posterior and anterior spinocerebellar tracts, as well as the cuneo-cerebellar tracts. Then coming also directly into the vermis is, I'm sorry, oh, for God's sake. Is visual, auditory, vestibular, and somatic sensory from the head proximal body. Then a second is the vestibulocerebellum, which you can see projects much more into the posterior lobe, right around the horizontal fissure. And what you have in here, as well as the floccular nodular lobe, and what you have is vestibular and visual inputs coming primarily in there. And these project down to the vestibular nuclei and make a control of balance, vestibular reflexes, and eye movements. So what you can basically see coming out of the vestibulocerebellum, including the floccular nodular lobe, is what? What you can basically see is the type of inputs that are going to come out into the medial vestibular nuclei. And if you remember, the axons of the medial vestibular nuclei form the medial longitudinal fasciculus, or MLF, that then does what? Projects caudally all the way down into the lower medulla and projects rostrally all the way up into the middle of the midbrain, where you're making a whole series of connections that basically is giving you up-to-date information about vestibular tone and visual tone, so one can now make particular eye movements and vestibular reflexes that are smooth. Then we have the so-called cerebrocerebellum, and what is coming in here is this is the kind of inputs that are coming out of the cortico-bulbar tract, and you're basically then taking the cerebrocerebellum and doing this reciprocal feedback between the cerebellum and the cortex to premotor, motor, and prefrontal cortices. So you're having the planning and executing of movements and the cognitive interactions with movements. So what you can basically see is that the cerebellum, in a much more gross kind of way, divides things in spinocerebellum, it's a somatosensory and proprioceptive, vermis into visual, auditory, vestibular, and somatic sensory, and then somatosensory input from the limbs coming in here. Okay. So now we basically start to look at a series of other of the deep cerebellar nuclei, and you can see with the, let me just here are the deep cerebellar. I should have rechanged these things. The 4 deep cerebellar nuclei, there are pairs. There's the vestigial nuclei, the vestibular nuclei, the globosus nuclei, and the emboliformis nuclei. They are in the vermis and the deep cerebellum. Then sitting out laterally, and we could see this in our sagittal sections, we can see that, again, wormy looking structure, sort of looking like that inferior olivary complex, very strange and distinctive looking structure. And those are the dentate nuclei of the cerebellum. So if we now basically look at this, we basically see the various types of inputs that go in. So we're going to have here are dentate nuclei outputs. Here are interpositive nuclei, which are globosus and emboliformis. Here's vestigial deep cerebellar nuclei. So here is how they basically come in. Association cortex goes through pontine nuclei, so the parietal lobe will project to pontine nuclei that then will project onto the dentate nucleus. And the dentate nucleus, if you remember, provided incredible amounts of input into the BL nucleus of the thalamus. And then the BL nucleus of thalamus then basically is modulating motor and prefrontal and premotor and parietal cortices. Where did I lose? Is my pointer right down there? Okay. Then we also then get inputs of somatosensory receptors on limbs and motor cortex. So here we're getting proprioceptive types of feedback. They are coming in through the spinocerebellar and pontine nuclei, and they go to the red nucleus and the red nucleus, which forms the rubrospinal tract, and the reticular nuclei that form the reticulospinal tract. And these interneurons are then controlling spinal cord modulation of either proximal or distal movements. Then we go to the vestigial nuclei, and we can see somatosensory receptors in the trunk, as well as auditory and visual information. These spinocerebellar paths come in primarily in the thoracic cord and some in the cervical cord, forming an output going to the vestibular nuclei. So you're getting a what? An idea of where your body is in space. Then we have another set of fibers that are coming in from somatosensory receptors in the neck, the labyrinths, and visual information. So now we're getting primarily cervical as well as vestibular, auditory, and visual information that then controls the vestibulocerebellar, goes to the flocculonodular lobe, and then projects to the vestibular nuclei to do what? Through the MLF, allow you to make the appropriate eye movements or postural movements depending on where you were originally in space. So we see all of these things right here. This is sort of a blow up. And now what you can basically look at with the cerebellum is you see the cerebellum has output via the dentate nucleus to the BL thalamus that then controls specific parts of the cortex. We then have fibers of the interposed nuclei controlling the rubrospinal tract, and then we see vestigial inputs providing inputs into the vestibular nuclei, as well as the reticular formation to produce what? Reticulospinal outputs. So what we basically end up with in this whole concept of coordination, and coordination, of course, involves what you're doing at any given particular instant in time that is dependent upon what you were doing immediately prior to it, and then doing the motor planning to continue doing things. So you're going to have motor and somatosensory loops. So at the spinal level, we're not only thinking of just sensory input, somatosensory input, and motor output, but we're also thinking of those two other factors, proprioception, which is what? The sensory events, the sensory activation, depending on what just happened prior. And then kinesthetic feedback, which is motor output that then basically loops in. We then see the parietal lobe playing a very, very important role in modulating output. And what you can see here is that the parietal lobe is going to have a lot of input coming from anterior thalamic nuclei, both the anterior ventral and dorsal medial nuclei, and then that then controls the anterior pontine nuclei. And those anterior pontine nuclei, of course, are going to start playing a role like the locus coeruleus, the dorsal tegmental area, that's going to be playing a role in certainly things having to do with attention, arousal, shifting of attention, shifting of arousal. And then what we have are prefrontal loops. Again, the AV nucleus of thalamus through the cingulate gyrus is going to be affecting the prefrontal cortex, the so-called executive branch of the brain in neuropsychology. And again, you can see that loop coming back into the anterior pontine nuclei that's either going to sustain arousal and attention. So when we think of the parietal lobe, we're basically thinking very often of things that we would necessarily call association cortex. So you're basically doing things like recognition, or activating motor areas that have to do with particular types of motor programs. And then the PFC, of course, is making decisions. Then we then look at the temporal lobe, and of course the temporal lobe, primarily through the ventral stream nuclei, as well as auditory contact, is modulating arousal and attention. And it too is getting input via the cingulate gyrus of the anterior ventral thalamus. Then we have the oculomotor control as to how we are going to basically control that vast majority of eye muscles that then allow us to make particular movements. And then, of course, the basal ganglia. So what we're looking at here, one of the things we're looking at right here is the basal ganglia. And what did you recognize when you finally got out of that striato-thalamo-cortical loop, or cortico-striato-thalamo loop, or thalamo-cortico-striatal loop, that one of the major outputs coming out of the globus pallidus is what? Going to the subthalamic nucleus, and look at what the subthalamic nucleus does. Again, it's feeding input into the AV nucleus of thalamus, that's further modulating this, as well as going to the anterior pontine nuclei that basically then allows one to maintain arousal and attention. And all of this stuff gets fed back into the cerebellum and then fed out again through the deep cerebellar nuclei, and the loop goes on and on and on and on and on. And there were the cerebellar nuclei. So again, just for your information, here are some further notes with the spinocerebellum. Okay, that we covered both in the talk and in the book. So that is the cerebellar nuclei. So let's see what we have here. Okay, it looks like a bunch of chats that happened a little bit earlier. Okay, why don't we take a short break? I'm going to re-warm up my coffee, and then we will go to the last so-called lecture on neurochemical neuroanatomy. So I'll be back with you relatively soon. What I will do is we'll go to the last of the lectures here. Which in a lot of ways basically summarizes and puts together a lot of the things that I've covered through the course. So very often when we moved into the area of neuroanatomy, by the late 1950s, it was basically thought that everything we ever wanted to know about neuroanatomy was known in terms of the structures. And of course, with the advent of electron microscopy, one could go and study synapses. So of course, any other major domination, if you go into the history of neuroscience, that when you basically looked at neuroanatomical structures, and then after the understanding of that, especially in the age of Cajal and Golgi and Lorente Deneau and a number of other prominent neuroanatomists, one then got into the area of neurophysiology. And with, of course, the work of Sir Henry Dale, the work of Sherrington, and whatever, you basically had the understanding of excitation and inhibition and the whole idea of how synapses work and the communication of information. So by the late 1940s and into the 1950s, it became very clear that we thought we knew everything we wanted to know about the neuroanatomical structures, but also wanted to know about how the brain communicates with each other in neurophysiology. But during this entire time, and by the way, when you think about that, you basically think of neuroanatomical structures and whatever, and then you think of the work of people like Papes and people and folks like that with the whole advent of psychosurgery, by basically arguing that there is localization, anatomical localization of function, and that if you go and ablate a particular part of the brain, you might be able to successfully alleviate something. And of course, you did that in terms of motor and sensory types of information. But then people got into more complex things and got into cognitive and psychiatric issues. And of course, we then got into a period called psychosurgery, where basically the idea is that a particular area with a putative function could identify things. So if you just think of the work that was found with the lesion studies in the 1930s of lesions of the septum and lesions of the amygdala, where you thought you were going to modulate aggressive behavior, you're going to modulate bad social behavior, and folks would work on that. So then in the and during that, of course, that time, when you basically looked at a large number of people that had psychiatric disorders, you have to realize that about 1950, virtually half of all hospital beds, and there were far more hospital beds then than there are today, half of those beds were occupied by long-term psychiatric patients who basically would be warehoused in the thing. And then in for early people in development, you would have large numbers of people with profound retardation or mild retardation who were basically institutionalized. And so what was very interesting there is that if you want to go and you want to do human experimentation in the early 1950s, you had 3 populations that were readily at hand. One were people in the military, 2 were people who were incarcerated in prisons, and 3 were people who were in long-term mental hospitals. So we basically go to, and obviously today you would never get through a human institutional review board with any of these studies, and the whole reason why we have institutional review boards is because studies like this were done. But in the early 1950s, people would use psychiatric patients, especially psychiatric patients who shared particular types of physiological symptoms that were indistinguishable from the group at large. So one major area was allergies, and that people would have allergies and would have a large mast cell response with histamine and whatever, and you would want to treat these people. So you would have hay fever season and problems with pollen and this and that and the other thing. So in any case, there was a French pharmaceutical company that basically was looking, they had a particular candidate drug that they thought would be a good allergy medication, an antihistaminergic type of drug, although they really didn't know that much about the way histamine worked, but they knew you got these mast cell activations. So what they started to do is identify people in psychiatric hospitals that had hay fever. And they basically gave the drug to these people, and they gave a placebo control. And when they finished doing their phase 3 trial, I think now everybody knows the difference between a phase 1, phase 2, and phase 3 clinical trial with COVID, they basically looked at the results. And what they found was that this particular drug was terrible for, it did minimal things on allergies. However, as they were doing these things, they would write down other types of side effects. And the nurses who were giving out the drugs and doing things and just looking at the notes of the people on the floor, they basically found is that what this drug did is it did nothing to allergies, but what it did was substantially reduce the hallucinatory behavior that a large number of the patients demonstrated. And then upon further looking, they basically saw that a lot of, although the delusions were not changed. Remember, a hallucination is a false type of perception, whereas a delusion is a false belief. So they started to now redirect that looking, and of course what they created was one of the first antipsychotic medications, which of course is chlorpromazine, otherwise clinically known as Thorazine. Now, the major thing about this drug is that you had to give it in very, very high doses. And basically what you would find now all of a sudden is this drug now got misused in psychiatric hospitals, because basically what you basically did was give the drug to sort of reduce all of the adverse psychiatric activity that would happen on a ward at any given time. And probably one of the greatest movies to show that was One Flew Over the Cuckoo's Nest, with such actors as Jack Nicholson and Danny DeVito, and then the famous Nurse Ratched. So if you I would always suggest that you watch that movie. And in any case, it was now thought that this drug had effects. Now, another thing that was being treated in the 40s and 50s was a large number of people with depression. And what now started to be used was the drug amphetamine. And during that particular time in the 40s and 50s, both the chlorpromazine and the amphetamines was thought to act through the major catecholamine of the time, norepinephrine. It actually works much more selectively, as we well know, on brain dopamine. But in any case, you started to see a yin and a yang, where depression or subarousal versus schizophrenia and a number of the anxiety disorders, which is hyperarousal, was thought to had to do with big increases in norepinephrine or big decreases in norepinephrine, and then the pharmacological tools to go and start to work at that. Where norepinephrine, dopamine, and other classic neurotransmitters were in the brain, they were known to be in the brain, but they really, the way you basically studied this was studying this basically outside of the brain, in what we basically call the sympathetic nervous system, that norepinephrine and epinephrine basically was there in the area of arousal and was thought to act peripherally. So slowly but surely, there was a shift from being that psychopathology or any kind of and remember, psychopathology was always under the domain of the Freudians, who basically believed that this was not a genetic difference, but this was an environmental differences between interactions between the child and the parent, particularly the mother. So a whole bunch of shifts started to happen in the 1950s. And of course, in the early 1960s, a series of techniques came into play that allowed us now to visualize neurochemicals in the brain and how those neurochemicals are then modulating activity in brain areas. So here are the classic neurotransmitters of norepinephrine, dopamine, serotonin, acetylcholine, and histamine. So in the norepinephrine pathways, what we basically can see here is that the large number of norepinephrine containing pathways can be found in from the lower medulla all the way up into the pons. There is a subgroup of norepinephrine containing cells in what is called the A10 cell group. That cell group up in the hypothalamus is primarily dopaminergic, but there's a small number of norepinephrine containing cells. So with the work of Dahlstrom and Fuchs, and then Ungerstedt, and then Lindvall and Bjorklund, what you were basically able to do is identify the norepinephrine containing neurons, and then found that these neurons had rather specific types of projections that then modulated a whole bunch of the functions that were attributed to them. So using the nomenclature of Dahlstrom and Fuchs, used the letter A to show you catecholamine pathways. So we go from caudal to rostral, and the most caudal catecholamine cell group is around the nucleus tractus solitarius, the A1 cell group. And you can see the more rostral one, other than the small one in the zona incerta, the A7 goes through the medulla and through the pons and right up to the pontine midbrain border. So the A1 cell group is found around the nucleus tractus solitarius, and the vast majority of these A1 cells project down into the spinal cord, and they innervate all three different parts of the spinal cord. One in the dorsal horn, especially in and around the substantia gelatinosa, where norepinephrine, when working on a presynaptic neuron in the substantia gelatinosa, can basically inhibit that presynaptic neuron. So as you know, for layer 2, the substantia gelatinosa, a large number of the incoming cells are nociceptive containing. They, when they fire, you get the sensation of pain. And these norepinephrine cells coming down from A1 and presynaptically synapsing in substantia gelatinosa shows you that norepinephrine activated in this way can inhibit pain. Then the A2 and A4 cell groups are primarily noradrenergic interneurons in the caudal medulla and in the more rostral medulla. Now, A3, A5, and A7 sort of form a continuum of scattered noradrenergic cells through the reticular formation of the medulla and the pons. And what they will basically form is eventually what is called the ventral noradrenergic bundle. So these are norepinephrine containing cells that are ascending. And notice where they ascend into. They ascend into the ventral midbrain. So what the ventral noradrenergic bundle will innervate is it will innervate the Raphe nuclei, so you can have a noradrenergic serotonergic interaction, and they will innervate the ventral midbrain, particularly the ventral tegmental area and the substantia nigra, so that the A3, A5, A7 ventral bundle will modulate dopaminergic activity of either the nigrostriatal tract or modulate dopaminergic activity of the mesocortical or mesolimbic tracts. The ventral bundle then continues to ascend throughout the hypothalamus and makes contacts in many hypothalamic nuclei, including the ventromedial hypothalamus, the lateral hypothalamus, the paraventricular nucleus of the hypothalamus, and they basically modulate an enormous number of functions. Also coming out of here is that these things will go up into such places as the amygdala and then the preoptic area. And the ventral noradrenergic bundle will then project up into the olfactory bulb, into the septum, and then into the cortex. So that is A3, A5, and A7. A1 is primarily descending, A2 and A4 are primarily interneurons. So what that leaves us is the A6 cell group. And the A6 cell group is morphologically identical to the cytoarchitectural characteristics of the pontine nucleus, the locus coeruleus. So all of these locus coeruleus and subcoeruleus cells will all contain and release norepinephrine. So the locus coeruleus then has a wide number of projections. One is it, just like A1, will project down into all three levels of the spinal cord, into substantia gelatinosa of the dorsal horn. It will also project into the ventral horn, where it will inhibit large alpha and gamma motor neurons. And where that comes from is primarily from a subgroup of locus coeruleus cells called the subcoeruleus. And here the locus coeruleus got into an awful lot of interest from people who had a psychoanalytic background. You might think that to be strange. But in a lot of ways, people were basically looking for a neuroanatomical correlate of Freudian theory. So one of the things of Freudian theory, of course, is dividing the personality into three parts: the id, the ego, and the superego. And we basically look at the id as primary process thinking, instantaneous gratification, and the whole idea of the pleasure principle. Whereas the ego was related more to a reality principle. It was still dedicated to getting what you want, but using sort of more realistic norms to get there rather than an impulsive act. So obviously, when one basically looked at this model of Freud and the whole idea of the id and having all of this unbelievable pressure to instantaneously gratify things, one of the other arguments that Freud would argue as a release valve for id-related issues, of course, was dreaming. And so it became very, very interesting that when one looked at the subcoeruleus and A6, that destruction of the subcerebrous and the A6 neurons could actually take an animal that was asleep and was in rapid eye movement sleep. That one of the things that goes along with rapid eye movement sleep is what? Some degree of muscular paralysis. And it was thought that the A6 cell group, by producing inhibitory effects in the ventral horn on these alpha and gamma motor neurons, was modulating that motor sort of paralysis that associated with the dream, and destruction of this area now allowed the animal to go out and start to move around during dreams. So of course, get into the whole idea of somnambulism and things like that, where you're basically acting out a dream. If you ever want to watch one of the greatest, funniest examples of this, there was a classic show on the famous honeymooners with Ralph Cramden and Ed Norton, the characters in Brooklyn. And Norton, who was a somnambulist, would be sleeping, all of a sudden get up. Of course, the guy had a sort of nightgown on, and he still had his hat on, and he would exit his apartment, walk upstairs one floor to the Cramden apartment, walk in, open up the icebox, and start to eat. Okay, so very, very funny kind of thing. But basically, the A6 cell group was thought to modulate that type of behavior. And you could see this interesting aspect of the locus coeruleus involved in attention and arousal. And then this modulation could sort of put together all of those components that Freud might want to see inside of a dream. Now, also, what the A6 cell group does is it interacts with the raphe nuclei all along the thing, will actually interact with the substantia nigra and ventral tegmental area. It will then reach up and produce inhibition in the cerebellum. And then it will provide input not only to the ventral noradrenergic bundle, but to the dorsal noradrenergic bundle, where now all of a sudden this dorsal noradrenergic bundle is modulating a lot of activity along the periaqueductal gray, and modulating the activity of the superior and inferior colliculi. The dorsal noradrenergic bundle now goes up into the hypothalamus, the paraventricular nucleus, and then makes a number of connections with thalamic nuclei. It then shoots up into all four lobes of the cortex and provides a major input into the hippocampus, so that A6 noradrenergic release results in noradrenergic release in the hippocampus that modulates a particular hippocampal electroencephalographic activity called hippocampal theta. And it is thought that hippocampal theta is associated with the consolidation of short-term memories. So, like I said a number of lectures ago, the A6 cell group is always sort of thought of as a cell group like the little engine that could, that given the relatively small number of cells, the outsized impact it has on spinal activity, etc. And also what the A6 and A1 cell group, when they descend into the spinal cord, do a third connection. not only with the dorsal horn in inhibiting pain, or the ventral horn in terms of inhibiting alpha and gamma motor neurons, but also around the pericannular gray, where the noradrenergic input goes into these pericannular gray neurons that release glutamate. And these outputs of the pericannular gray now go out into the sympathetic ganglion. And the sympathetic ganglion, again, is a repository in the peripheral nervous system that contains a lot more epinephrine. And then the sympathetic ganglion will release epinephrine and norepinephrine based on that glutamatergic synapse and into the blood supply. And right nearby is what? The adrenal medulla that sits atop the kidney. And the epinephrine and norepinephrine secreted by the sympathetic ganglion acts on the adrenal medulla to release a quantal increase more of epinephrine and norepinephrine. And of course, what I'm describing for you here is the sympathomedullary noradrenergic system, modulating a whole host of stress-related types of responses, having to do with respiration, having to do with cardiovascular, having to do with pupillary dilation, and having to do with a whole bunch of peripheral activity. So we can see that A6 cell group does an enormous amount of activity in a number of different areas of the brain, and it's doing a lot of its activity when very often we are either aroused or attentive and awake, but we're also, it's doing activity when we are asleep. Then what we have is the dopaminergic cells, and we've gone through this before. And what we basically see here is the substantia nigra contains two different types of cell groups. One is called the A8 cell group, which is the substantia nigra pars lateralis, and then the A9 cell group, which is the substantia nigra pars compacta. And these form the nigrostriatal tract that will primarily project to the caudate nucleus and the putamen, and then the caudate nucleus and putamen will then give its outputs upon the globus pallidus. And what we have seen before in the striatum lecture is that the amount of dopamine release in the nigrostriatal system will activate either the D1 mediated direct pathway of the striatum or the D2 mediated indirect pathway of the striatum. Then what we have is the A10 cell group. The A10 cell group originates in the ventral tegmental area. Remember that the number of dopaminergic neurons that are found in either the substantia nigra or the ventral tegmental area are about 50% of the total number of these projecting neurons. So what does this pathway do? The ventral tegmental area will project through a mesolimbic system to the amygdala, to the accumbens, to the hippocampus, to the septum, and then to the olfactory nuclei and the olfactory tubercle. The mesolimbic area of the mesocortical projection of the ventral tegmental area goes up into primarily the frontal lobe and the neocortex. So that is A10. Like I pointed out, A11, which basically sits in an area called the zona incerta, is also very rich in dopamine. And the zona incerta is in the area around the fields of Prell. Remember those major outputs coming out of the globus pallidus as they move out to the thalamus. So inserted within there are a whole series of predominantly dopaminergic, but also a sprinkling of noradrenergic cells in the diencephalon. The vast majority of these cells actually reach and go all the way down into the spinal cord. And again, their major destination is into the dorsal horn of the spinal cord, where they will inhibit noxious input. So dopamine can work in inhibiting pain by via this spinal direct pathway. Then there is a last set of dopaminergic or catecholamine fibers, which is basically found down here in the base of the hypothalamus, right around the median eminence zona externa. And that is the A12 cell group. And that A12 cell group is called the tubero/infundibular dopamine pathway. So a tubero, you're sort of sitting along the base of the brain. Infundibular, you're right around the infundibulum, although you're not using the posterior pituitary. What basically happens is that these dopamine-containing cells release dopamine into the hypothalamo-hypophyseal portal system. So we are basically using these cells are part of the adenohypophysis. That is brain cells that are modulating the anterior lobe of the pituitary. So the dopamine that's released here goes through the hypothalamo-hypophyseal portal system into the anterior pituitary. And what it acts on are cells that are able to release the hormone prolactin. And of course, if we basically think about prolactin is found obviously in both men and women, but the major thing that we basically think of for prolactin is the release of prolactin in lactating postpartum women that allows for the milk ejection reflex. However, prolactin has a number of other kinds of hormonal types of effects. And so what does dopamine do? Dopamine released from the A12 tuberoinfundibular region will act to inhibit prolactin. So now we get a very, very interesting thing, that very often, if one wants to see the efficacy of dopamine blocking drugs that that might be used for the treatment of severe psychopathology, very often what will happen is there will be blood samples taken to look at prolactin levels. And because when the drug is given, let's say orally or given via injection, it's happening globally. And of course, anything that's acting on the A10 cell group or the A8 cell group or whatever is also acting on receptors of the A12 cell group, which is found in the anterior pituitary. So under normal circumstances, dopamine in the anterior pituitary is inhibiting prolactin expression cells. So now when you give a dopamine antagonist, and you can see what you will now start to see is hyperprolactinemia. We are not saying here that prolactin is involved in extreme psychopathology. We're just using the prolactin release as a marker for the efficacy of the dopamine antagonist. So now I've covered the 12 different major catecholamine cell groups, with A1 to A7 being primarily norepinephrine, A8, A9, A10, and A12 being dopaminergic, and A11 having a small amount of norepinephrine, a larger amount of dopamine. So now we go to the serotonin pathways. And again, this is sort of a review. We're putting things together in terms of the Raphe nuclei. What we basically encountered was the Raphe nuclei in the very caudal medulla, and we could see the Raphe nuclei go all the way forward into the middle of the midbrain. And basically what we have, and this is not a very good drawing, you have the medullary Raphe nuclei here, you have the pontine serotonin nuclei here, and then you have the midbrain nuclei here. So what we basically see in the rat and the cat is that there are 9 Raphe nuclei marked as B1 to B9, and where B1, 2, and 3 are medullary Raphe nuclei, 4, 5, and 6 are pontine, and 7, 8, and 9 are mostly in the midbrain. And again, only about 1/3 of Raphe cells that are identified cytoarchitecturally contain a neurotransmitter serotonin. Also, serotonin is a wonderful example of a system in which there can be co-release. So we basically saw identification of a cell that not only releases serotonin, but also releases substance P and releases TRH, 2 short peptides. And so what we basically see is that Dale's law is not in effect. So when we look at B1, B2, and B3, and they include what we call in the rat, the nucleus Raphe magnus, what we basically see is descending control. So these Raphe nuclei are getting what? Lots and lots of lateral spinothalamic input. So a lot of that can be nociceptive types of input. So therefore, when this nociceptive excitatory input comes in via the lateral spinothalamic tract into the Raphe can then be activated and work in not only the spinal cord, but the spinal trigeminal nucleus, especially around the substantia gelatinosa, whereas what serotonin is doing is it's acting on substantia gelatinosa cells, it's strongly inhibiting them, and therefore acting as a pain inhibitor, basically saying, hello, we got the message, stop sending, okay? And you do that not only in the spinal cord, but also the spinal trigeminal nucleus, which of course is innervating the head. Then what we see for the pontine raphe nuclei, they not only project up into the cerebellum, but they basically control things like the locus coeruleus and deep reticulus that contain acetylcholine. So here is another interesting kind of thing. Think about two types of peripheral arousal. We have the sympathetic nervous system and we have the parasympathetic nervous system. So in the sympathetic nervous system, we have noradrenergic arousal. In the parasympathetic nervous system, we have two transmitters at play. One, which is serotonin, which will mutually inhibit dopamine, norepinephrine neurons, and they will, and the serotonin will also act on another parasympathetic modulator, acetylcholine. And very often we see acetylcholine as part of the parasympathetic nervous system to do what? Post-ingestive digestion, peristalsis, etc. Okay, so we see these pontine raphe nuclei sort of mark B4 to B6 doing these parasympathetic modulatory effects. Then we have the B7, B8, and B9, where we're looking at these midbrain serotonergic fibers. Some of them, particularly in B9, literally project back to the raphe magnus and enhances their effect. So a serotonin to serotonin interaction happens here to promote pain inhibition. But look at what they also do. What these midbrain serotonin neurons modulate the substantia nigra and the ventral tegmental area. They modulate both the auditory and optic tectum. They modulate the so-called dorsal dorsal limbic system, the habenula. They modulate the thalamus, the hypothalamus, the basal ganglia, the hippocampus, and then the whole limbic system. And then again, serotonin will reach forward into the neocortex to modulate effects. So now we go to a fourth system, the cholinergic pathway. And basically, whereas there are, the way we have basically looked at the cholinergic pathway is by the activity of acetylcholine esterase. And acetylcholine esterase is the enzyme that breaks down acetylcholine in the synapse. And one could basically see acetylcholine esterase in all of the, virtually all throughout the brain. But then it was finally figured out that acetylcholine esterase, in addition to breaking down acetylcholine, has other activities. So therefore, it took work using immunocytochemistry and looking at a precursor of acetylcholine, choline acetyltransferase, CHAT, and doing immunocytochemistry, and then realizing that the number of cholinergic cells in the brain are actually quite less distributed. They're not all over the place. So what we have are two major areas for the cholinergic pathway: a caudal area and then a wide swath of a rostral area. So the caudal area is found in the laterodorsal and pedunculopontine tegmental nuclei. So they're basically sitting in the rostral pons and the very, very caudal midbrain. So these acetylcholine containing cells, and some of these cells are those very, very large NRGC cells, nucleus reticularis gigantocellularis cells. So those cells contain acetylcholine, and through a bifurcating axon can reach all the way down into the spinal cord and then can go up into the brain. But a number of other cholinergic cells have projections of this caudal group that controls the deep cerebellar nuclei, usually through excitation, activation of the superior and inferior colliculi, activation of the medial habenula, activation of the thalamus, activation of the substantia nigra, then modulating the locus coeruleus, pontine reticular formation, vestibular nuclei, raphe nuclei, and medullary reticular formation. And in virtually all of these cases, this activation through cholinergic neurons is excitatory. Then what we basically see is a swath of cells starting in the very basal forebrain in an area called the nucleus basalis of Maynard. And then those cells sort of reach out in an arc into the caudate nucleus and the putamen and the nucleus accumbens. And then that arc continues out past the amygdala into the medial septum. Now, the first two areas that I'd like to very much talk about is the nucleus basalis and the medial septum. Both of these are cholinergic containing. They're a tight set of cells. And then there is cholinergic innervation of all four lobes of the neocortex, as well as of the neocortex and the olfactory bulb. And what you're having is cholinergic activation of the neocortex and the olfactory bulb by the nucleus basalis of Maynard. And the nucleus basalis of Maynard very often is activated by alertness and arousal coming from noradrenergic activity. So all of a sudden, what the nucleus basalis is doing is providing excitation into many different parts of the neocortex during periods of time that may be high in arousal or high in selective attention. So what became incredibly important was the work of Joseph Coyle in the early 1980s. He was another student of Solomon Snyder, and he basically determined that in both humans and in animal models, there was a selective loss of nucleus basalis cholinergic cells. And with that loss, the amount of that loss was associated with the severity of Alzheimer's disease. So basically, this and a series of other studies in the early 1980s basically formed, just like you had in the early 1980s, the dopamine theory of schizophrenia. Here you started to have the cholinergic theory of Alzheimer's disease, with the basic loss of nucleus basalis cholinergic tone. Further, what we basically know is that on the other area of this rostral cholinergic pathways is the medial septum. And the medial septum has a set of fibers that what is going right by here. What is going right by here is the fornix. So the medial septum will provide cholinergic input into the cingulate gyrus that will join the input that the AV nucleus of the thalamus provides for the cingulate cortex. So you're activating this part of the limbic system. In addition, the medial septum sends cholinergic cells out to the amygdala, which of course is mediating things like fear, et cetera, and you are then having medial septal input going via the fornix in the opposite direction. Remember, the large part of the fornix is hippocampal outputs coming out of the fimbria, coming into the fornix, and then going through that whole loop and eventually finishing in the mammillary bodies. But as the fornix moves through, and we have the columns of the fornix here, we actually have not only a hippocampal efferent, the fornix, but what we have is a hippocampal afferent, the septohippocampal pathway, which will enter through layer 6, the alveus. And again, what basically was found was that loss of cholinergic tone in the medial septum would produce some of the shorter term memory difficulties and whatever that we see with cholinergic innervation of the hippocampus. So those are the cholinergic pathways. So now we then go into other classic transmitters. So one was, of course, the histamine pathway. And what you basically see with the histamine pathway is one very large nucleus that contains histamine, and that basically projects to all the different areas of the brain. So histamine in the brain is synthesized and released by the tuberomammillary nucleus. And you can see it innervates the olfactory bulb, the striatum, the cortex, the hippocampus, the hypothalamus, the thalamus, the tectal nuclei, the cerebellum, and then all the way down into the brain. So in the mid-1970s, one thing we also knew was that with neuroclassic neurotransmitters, what do we have? We have basically with the histamine, with acetylcholine, with serotonin, with dopamine, and with norepinephrine. All of these are derived by what? Basically enzymatic changes in a single amino acid. So we have the single amino acid tyrosine giving rise to norepinephrine and dopamine. We have the single amino acid tryptophan giving rise to serotonin and melatonin. And then we have the single amino acid choline giving rise to acetylcholine. And of course, by the early 1970s, it was then appreciated that there were three other single amino acids, glutamate, glycine, and GABA, all of which acted on not a simple receptor, but a simple receptor complex to provide excitatory effects or inhibitory effects. But all during this time, we also knew in the brain that there were very simple peptides in the brain. And folks never gave them very, very much of a attention in terms of functional activity. This, of course, changed with the identification of the opiate receptor. And the opiate receptor, of course, was triggered by Richard Nixon's war on drugs and the creation of a place within the National Institutes of Health called the National Institute on Drug Abuse. And what was interesting is the third grant that was ever given out by NIDA. The third one was given to Solomon Snyder, and Solomon Snyder, with his then graduate student at the time, identified the opiate receptor 4 years later. Another NIDA recipient, Eric Simon from NYU, did the same thing, and then a Swedish guy named Lars Terranius did also the third thing. So now, all of a sudden, we identify opiate receptors that are extremely sensitive to opiate agonists and antagonists. And then 2 years later, a Scottish team using costolates identified 2 pentapeptides as the primary things that bound to opiate receptors. And all of a sudden, the doors flew open, because very, very quickly thereafter, people took other, what they considered active peptides, and found receptors in the brain for those things. And we then developed an enormous panoply of neurochemical tools that act on receptors that basically control synaptic activity, etc. So we blew the doors off. So it's very funny. We don't sit there and count. If we count every particular peptide and whatever, we can then have probably 500 to 1,000 active substances. But very often we talk about these things in terms of families. And some of the peptides may share certain characteristics and bind to particular receptors and whatever. So an opiate receptor is bizarre, because what it can do, just think, let me jump back a second. We see norepinephrine and dopamine, okay? What's the difference between norepinephrine and dopamine? Dopamine is a precursor to norepinephrine within a cell. What that cell needs in order to produce norepinephrine is dopamine beta hydroxylase, one little enzyme, and we change it from dopamine to norepinephrine. Yet, if we take dopamine receptor cell cultures and bathe it with norepinephrine, the affinity of norepinephrine to bind to those dopamine receptors is extremely poor. The ability of dopamine to bind to those receptors are very often 1,000 to 1,000,000-fold greater affinity. So this infinitesimal change in one enzyme that creates norepinephrine from dopamine results in an enormous difference in terms of the binding characteristics. And by the way, it's vice versa. If you have noradrenergic receptors, they bind dopamine, but at far less affinity, and you need far more of the dopamine to act there. However, when you look at an opiate receptor, let's take the mu opioid receptor. The mu opioid receptor binds enkephalins with great affinity, But the delta receptor binds enkephalins with even higher affinity. So there's two different receptor subtypes. But then we come along with a peptide, beta endorphin, which is 31 amino acids long. And it will bind with equal affinity to the mu receptor like enkephalins, whereas it will bind much poorly to the delta receptor. So what I'm telling you is a new opiate receptor will bind a 5 amino acid peptide and a 31 amino acid peptide if they happen to be enkephalins and endorphins. Okay, so it's very, and basically, you don't venture out now, but if you were to venture out into Brooklyn or Manhattan on the west side, and you basically see ships come and leave docks, you always use docks as a receptor. It's a difference between binding with a rowboat and binding with an ocean liner in terms of the sizes of these things. So all of a sudden we have a whole series of peptidergic pathways. So one pathway, and this is very interesting, is because very often what we would basically do is divide things into three different categories arbitrarily. We had neurotransmitters, then we had releasing and inhibitory factors, and then we had pituitary hormones. And very often we would say that the three are distinct from one another. The pituitary hormones should only work out there in the periphery. The releasing and inhibitory factors should only work on the anterior pituitary, and the neurotransmitter should only work in the brain. Well, I already blew that up by telling you that dopamine is a neurotransmitter, but it is also an inhibitory factor by inhibiting prolactin. So now we can basically look at corticotropin releasing factor. And corticotropin releasing factor, we see really comes out of one particular brain nucleus that I talked about when I talked about the hypothalamus, the paraventricular nucleus of the hypothalamus. And specifically, we are basically talking about the PVN, the parvocellular division of the PVN. Remember, the magnocellular division of the PVN gives rise to those axons that will form the infundibulum and go to the posterior pituitary. The parvocellular division, they have CRF-containing cells. A subset of those CRF-containing cells, mostly in the ventral parvocellular division, basically come down to the zona externa of the median eminence and release CRF into the hypothalamo-hypophyseal portal system, and where it then activates adrenotropes in the anterior pituitary to release ACTH that then acts on the adrenal cortex. And then the adrenal cortex releases corticosteroid, cortisol or corticosterone, which will produce both long loop inhibitory feedback on the PVN or short loop inhibitory feedback on ACTH containing cells in the pituitary. But in addition, CRF is released from the more dorsal parts of the parvocellular PVN, where it will innervate the lateral hypothalamus, the amygdala, the bed nucleus of the stria terminalis, the septum, the thalamus, and then the dorsal raphe, and then down into the locus coeruleus. And basically, CRF, there are two types of CRF receptors. A CRF1 receptor that has the effects of arousal, anxiety behavior, disruption of sexual behavior, disruption of sleep, simply because the animal is in a sympathetic effect, that very immediate effect of CRF. That CRF is an alarm clock, boom, activating what? Whereas the noradrenergic system via the nucleus tractus solitarius or via the locus coeruleus is activating the sympathomedullary branch of the stress system. The CRF is activating the adrenocortical branch. And you then have a whole bunch of brain structures that are activating through CRF1 receptors all of these short-term stress type of effects. But there are also CRF2 receptors that appear to be involved in slow adaptive recovery and appetite suppression. So all of a sudden, what don't you want to do when if something is hunting you and wants to eat you? You shouldn't be standing out there eating grass or something. You should be getting the hell out of the way. And then with the the CRF2 receptor does that, but it also works in slow adaptive or longer term effects of stress. Then what we have are opioid peptides. And opioid peptides, most of the opioid peptides that we find act as interneurons. They act in rather specific types of things. So there will be opioid peptides in where? The caudate and the putamen. And we saw that when we looked at the caudate and putamen, and we looked at the direct pathway. So where dopamine binding to D1 receptors produces excitability and will act on the release of enkephalins from the caudate and putamen down to the globus pallidus. We can also see opioid peptides in the accumbens, in the septum, in the amygdala. The one area where we see a rather large and long pathway emanates from the arcuate nucleus of the hypothalamus. And the opioid pathway that we're talking about here is a pathway that releases beta endorphin. And what you can see here is that when this beta endorphin pathway is released, it will activate the periaqueductal gray, it will activate the ventral tegmental area, and then it will go down and activate a bunch of pontine and medullary nuclei. So what we basically see here, by and large, is most of the opioid peptides are working as interneurons in the periaqueductal gray, in places like the locus coeruleus, the ventral tegmental area, and then in the raphe nuclei that are further down into the medulla. And the other interesting thing is you can see that these opioid peptides are co-localized with one another within hypothalamic nuclei. So here, as I told you, POMC, this pro-opiomelanocortin, which is the precursor of beta endorphin. Beta endorphin is the C-terminal fragment of POMC. You can see where these POMC cells are in the arcuate nucleus of the hypothalamus. Here's our friend, the third ventricle. And then what you have is the mammillothalamic tract, and you have the fornix. So you can see where you are. This would be the ventromedial nucleus up here. And now what you notice is that there's an overlap of these cells that contain the shorter peptide in opioid enkephalin. But these enkephalinanergic neurons don't really project anywhere. They act as interneurons. Then we have a third opioid peptide, 17 amino acids long, called dynorphin. Notice the kind of words that got used, because first came the enkephalins, then came endorphin, endogenous opioid, then came dynorphin, very strong opioid. That's how it's sort of described in 1979 by Avram Goldstein. And you can see the dynorphin is sort of much more in the periventricular zone than in the arcuate zone. Then finally, an orphan opioid peptide called nociceptin, which was not identified immunocytochemically, but rather identified when the opiate receptor, the opioid receptors were cloned, and the opioid peptides were cloned, and we discovered the fourth class, nociceptin. And there really aren't that many here, but it's more ubiquitous. So now let's look at the GABAergic interneurons. And what we can see is that most of the GABAergic interneurons are found right within particular areas. So there's a large amount of GABA interneurons found here in the neocortex, in the hippocampus, in the superior colliculus, in the pons, and in the cerebellum. And then what you can now see is that there are two longer looped GABAergic neurons, one coming from the hypothalamus up to the cortex, and then the second one that we have described over and over again when we were comparing Parkinson's disease with Huntington's chorea, that what we can see is that GABAergic neurons originating in the striatum will go down and inhibit the globus pallidus, but they will also reach down into the substantia nigra, pars reticulata, to inhibit the dendrites of substantia nigra, pars compacta cells that in turn send dopamine ascending projections to the striatum. So that is sort of a overview, and I can probably go on for another 6 lectures to go on about all of the different transmitters and where they sort of get where they do things, where there are multiple receptors and whatever. And by the way, this type of work has is just continuously nonstop. So you basically see with innovations, what was thought to be, we knew it all in the 1950s because we knew a lot more about the structure and the underlying neurophysiology of the brain. We then started to discover the classic neurotransmitters. We started to discover the excitatory and inhibitory amino acids and where they go. And then we discovered the world and universe of peptides and where they go and where they interact. Okay, so I'm hoping that this lecture today and the lecture at the end of last week with the cortex fills in some of the other gaps that you have for the sort of big questions that I asked you on part 2. So at this point, what I'd like to do is open things up to any kinds of questions that you might have for in terms of the exam. If you have any remaining questions about the paper, and then if there's anybody here that has not made an appointment with me for taking part 1, we can go and we can do that. The other thing today that I sent out was an announcement. I get announcements. I'm assuming you all get announcements. If you don't, then Queens College has failed terribly. But they basically sent me an update that basically said, here are the courses you teach, Bodnar. Here are the people in the class. So there were 19 of you that were registered through Queens College. And then I saw, as of today, none of you handed in a course evaluation. I look at these course evaluations, and certainly this year, I would look incredibly at these course evaluations, because even though I've taught this course now for over 40 years, this is the first time I've taught it online. And I know we have gone through a whole bunch of bumpy things that have gone on, but hopefully there have been some good things that have gone on. And it would be of help to me if you gave me comments as to whether or not, if I have to do this again next fall remotely, how I could do it differently or better. One, I know I could do it better by not having one of my lectures all of a sudden disappear because I had a power outage, and I had to use today as the makeup time. Okay, so I open it up to all of you.
I was going to say, that's kind of weird because I did all evaluations on Tuesday.
You what?
When I finished, they just...
You're breaking
I did all
up.
the evaluations.
Yes.
Crap.
Okay. So if you sent in an evaluation and they didn't receive it, send it in again. I just don't know what to say. So you did the evaluations on Tuesday. I got that email on, maybe they were off by a day. I don't know. Who knows? Okay. Anybody else?
I actually tried to leave an evaluation, but couldn't log into the thing. It doesn't recognize any of my QD passwords.
Oh, now,
I'll try
in that message, in that message, if you read all the way down at the bottom, there's some link to IT, so you should basically tell them. So she's saying register with CAMS.
Okay, all right, thank you.
Yeah.
By the way, you know, you should know in advance that out of all the professors, you are the one that has nailed it on video delivery of teaching.
Oh, that's a nice thing for an old fart to know.
No, well, you might have needed.
I mean, I'm thinking of all the rest of my fellow faculty members. I can't think of a person who is less technologically adept than I am.
But
like
that's an advantage in your case, because you have total you have no misconceptions about your technical abilities. Therefore, you allow other people to fill in where they don't reach, whereas others are trying to I mean, maybe they know it's the Dunning-Kruger effect, you know. Sometimes it's been the smoothest by far, and when it comes to just the way that you have made it possible to learn despite having the class together and all of the extra stuff that comes in, you clearly want us to understand. You clearly want us to learn. So there's some that part comes through video, or I'm sure just as much in person, where it's absolutely clear, and it's actually motivating to do the work for you because you want us to understand and you want us to get it. It's not, you know how challenging this course is, and you've also used the way that you deliver is neuroscientifically informed, right? So you're clearly taking advantage of the knowledge that you have to deliver it in a way that our brains will respond to, and you're the only person doing that. Like it's crazy to me, but so I want to make sure all of that gets into a review. So I'm going to try and get back into the system. I don't know why it was so difficult, but I'll try and get all of that down in words, because it's been it's kept me going throughout the semester anyway, when other things have been harder to keep a hold of. So thank you.
Well, thank you. But the major thing I'm really hoping that you're able to do on top of, I realize, look, we're talking about our brains. I'm sorry, it's complex. If it wasn't, we wouldn't be having this course, because we wouldn't be doing any of this kind of stuff. It's because of our brains and because of its complexity that we're able to have a course like that. And one of the other things that I'm always hoping is, I find this to be a fact-based course. I find this to be able to tie structure and function together. And then the other thing that I find that I hope, and again, I'm terrible about this in terms of spatial, so that you actually see the pathways in some way, shape, or form, and you see this, and especially now as we continue to make all these incredible advances with brain imaging and things like that, that you don't have to take somebody else's word for it. You can literally look at the things and see this and identify the things for yourself. And then, it's a language, and it's a language that you're going to use with what? All of your scientific colleagues in whatever particular field you're in, and then with other collaborators, especially for those of you who move into more clinical areas where you're going to be talking with a rather wide kind of audience about looking at this. And that's what my goal is. And I always say, you started graduate school, and maybe someone has never said this before, but when I feel like when you start graduate school, you're learning things, hopefully that either are new or that are now built up, that you're going to hold and use for the rest of your career, wherever that career brings you. And so that becomes the important thing. What I hope you don't do is take your notes and take your book and close it, and close that part of your mind away, and not use what we learned here in this course in terms of further elucidating things that you may come up in other courses. And don't be surprised if when you then say, aha, I learned this in Bodnar's course and I'm taking this now, and maybe there's a connection. When you talk to the instructor who says that, they may not know that. You may know more than they do. I don't know. I would hope that anybody who's in neuroscience would take some kind of neuroanatomy course. You have to know how these things are structured and connected and whatever. Okay? Okay, so is there anyone else or any other kind of issue? If you have so, okay, so people are writing me. How am I going to say? Going to send in the paper. I asked for it to be done, I think, by the end of today. I said around the 10th. And then I got from somebody, I forget who I talked to, one of you, it might have been during the exam, and I said there's a panic as to whether it was due at 5 o'clock today or midnight today. Then some people asked, can I do it tomorrow? One person asked, I have very busy time, can I do it by Sunday? Let me just tell you, get the paper in by the middle of next week. I'm not giving you a deadline or something, but what I want to do, as soon as I get all the grades together, I can put them all together. I can then give out grades, and you'll be finished with this whole thing. So nobody's going to get penalized for this, but maybe even just send me a brief note in an email that says it's a little delayed and it's coming. And then, like I said, for part 2, I will release the question somewhere this weekend, and you'll have all the way to next weekend, the 18th, to answer them and send them back to me. Now, if shit happens and you can't do it, and you say, I need till Sunday to do it, just write me an email and tell me that. And, I recognize the unbelievable amounts of stress. I'm under a lot of stress, the amounts of stress that you guys are under, that you have to put all these things together. But what I'm hoping is, and I gave one last exam because the person had to do it on the first moment on the 21st, and I'm hoping by the end of the 21st, I'm able to submit your grades, okay? So there is the, so if you give me all your stuff by the 18th, I can read it over that weekend and get through it. By the way, a number of you, when you send me a paper, I read the paper and I send you the input right back, okay? Okay then. So if there is nothing else, have a very good luck with the rest of the course, good luck with the rest of your exams, and somehow have a nice holiday. Bye-bye.
Bye. Thank you.
Thank you.
Thank you.
Bodnar.
You're the best professor.
Thank you.